progress. Recording stopped.
How's everyone doing today?
Good.
Good? It's nice to have a little bit of nice weather this week. Thanks, Musa. I'm just getting myself set up here. Is Chip on there yet? Oh. He's there?
Yes.
Hey, Chaitan.
Hello.
Hi. Hey, could you turn on your camera just for 1 minute so we can see you? I don't think I've ever even seen you.
Oh, okay. All right. Hi, can you
Oh,
see me?
yeah, hi.
Hi everyone.
You can see everyone? Okay, good. Yeah, well, I just wanted to see your face. Have you guys seen him before?
Yes.
Some of you have, some of you have not. So you know, go, just so he remembers who's here.
I am Muslim. He knows me. He's my lab mate.
Ah, okay, so
Both
you
went to the same-
talk with him a lot on Zoom and stuff?
Not lately, but I have talked to him multiple times.
Okay, alright.
Hi Chetan, I'm Vivek.
Hi Vivek. Yeah.
I work with Dr. Lauren.
O'Donnell, yep.
I'm Matt. I'm part of the pharmacy program, but I'm working in Dr. O'Donnell's lab this year.
Huh?
Okay.
Hello.
Nice to meet you.
I'm Melanie. I am also a new student, like you.
And whose lab?
Dr. Bay's lab.
Dr. Bay, Misooks, okay.
Hi,
Great.
Chetan. I'm Afsana. I'm at Dr. Widender's lab.
Okay. Hi Afsana.
Hi, my name is Yong. Nice to meet you. So I'm your the AAPS the mentor. So you should reach me out once you are on campus so that you
He's
can
your
settle down.
AAPS mentor, okay, yeah, you better reach out to him.
Definitely. Yeah.
And he works with Dr. Bay. And then it's me,
Okay.
Dr. Kavanaugh, hi.
Hey.
Okay, you can turn off your camera again. I just wanted to see your face, and I didn't know if everyone ever got to introduce themselves. So, okay. So today we're going to finish, well, we're not going to finish. We're going to continue talking about GTP, binding proteins. What else are we going to do? Talk a little bit about adenylate cyclase, different isoforms of adenylate cyclase, and I don't know how far we'll get. And then some things about phospholipase C. So we're just adding more detail to the G protein coupled signaling pathways. Rather than just knowing the basics, we're just diving in a little bit deeper here today. Okay, so let me see if I can share my screen. You should have some handouts on your canvas. I'm just gotta set them up here. Just give me a minute. Okay. Move those out. Bless you. Let me just find where they are on my computer. I'm getting there. Just give me 1 minute. Okay, here we go. All right, so try and get there. Okay. Where's your course? Where's your course? right, so we already went through this stuff. We were talking about the alpha subunits. That should be alpha subunits. The sites on the alpha subunits that interact with the membrane and other proteins. We talked about the C-terminal domain. Is where it dictates how specific the alpha is for the receptor. We talked about the N-terminal domain that interacts with the beta-gamma subunits. Right? And then we talked about this SW1, 2, and 3 domains that undergo conformational change that allow the exchange of GDP for GTP, right? And it also facilitates the binding to the effector enzymes. And then it has all these, G proteins have these domains that we call the RAS-like domain, and these are similar across all the alpha subunits, and this contains the GTP binding site, binding surfaces for beta-gamma subunits, receptors, effectors, and the regulator of G protein signaling proteins. And then we talked about the helical domain. It's more variable. It covers the GTP binding site and undergoes conformational change to allow GDP to dissociate and aids in GTP hydrolysis. And then we talked about the different beta-gamma subunits, and we did a deep dive into that where you all presented different diseases or drug mechanisms that interact with the beta-gamma subunit, right? So I don't think we finished quite this part. I don't think we talked about the where's my notes on this? I don't think we looked at table 4.3 in the book. Let me just go there a second. Right, yeah, so this is G protein receptor kinases. And what they do is they phosphorylate here. So we'll write down. Phosphorylate the C-terminal regions of the G protein coupled receptor. So these kinases, and then in your book, and I'll write it down for you, on page 98 or in the chapter 4 handouts that are on your, I think it's chapter 4, on chapter 4, part 2, right? Yeah. You have table 4.3 in your notes. And so it shows you that there's at least 7 different subtypes of or this G protein receptor kinases, or these GERCs. They are expressed somewhat differently, where you have GERC1 and GERC7 only in the retina, and then GERC4 in the testes, cerebellum, and the kidney, and then the rest are ubiquitous. They have some similar structures with a kinase domain, a homology domain, and a variable C-terminal membrane targeting region. So just to know that there's 7 subtypes, GRK 1 through 7, GERC 1 and 7 only in retina, and then GERC 4, testes, cerebellum, and kidney. And then the others, 2, 3, 5, and 6 are ubiquitous. And so by phosphorylating this region of the receptors, they can either activate them or desensitize them, right? Okay, and then finally, in figure 414, look at that figure. It talks about how PKA and these GERCs can desensitize the receptors by phosphorylation. So they call A heterologous. Thank you. I just had to get my tongue to stop moving. Heterologous. If I stop myself and close my eyes, I can say it. Heterologous desensitization. So what happens here, it tells you here, under mild stimulation, the generation of cyclic AMP results in the phosphorylation of all proteins, including receptor molecules, having the PKA substrate consensus motif. So basically, cyclic AMP activates protein kinase A, right? And then the phosphorylation of the receptor in this case is going to occur regardless of whether they're occupied or not. And so what happens then when PKA phosphorylates the receptor, you get a generalized downregulation of all the receptors that regulate cyclic AMP production. So this leads to a general desensitization of all receptors that generate cyclic AMP. So cyclic AMP activates. So this is like a feedback, right? Then a feedback inhibition of the receptor. My notes for this didn't turn out. Okay, anyway. And then for GERK2, this happens only under strong stimulation, where the beta gamma subunits associate with the receptor. They act as an anchor for this GERK, and that phosphorylates only the attached receptor. So it's only going, this is not going to be as a widespread desensitization. This is going to be a more specific desensitization. Okay. And this one's going to depend, this one's going to require strong stimulation, right? Okay, so this pretty much ends talking about the trimeric G proteins, talking about the alpha subunit, how it's attached to the receptor, how the sites that are important for GTP exchange, GTP hydrolysis, the beta gamma subunits. We now know they're more important than what we learned originally. They have a lot of function and they're good. They can be good targets for certain diseases. And then talking about regulation of the G protein either by these GERCs or in this case by desensitization by a different kinase. So then that's all the trimeric GTP binding proteins. But we also have these RAS proteins that are monomeric proteins, right? These do not have the three subunits. So they are monomeric, and they basically are mostly related to the alpha subunit of the G proteins that we talked about. Okay, and RAS proteins are generally thought of when you think of as cancer. When you think of cancer, because we'll talk about that, but they're expressed in many different types of cancer. RAS, these proteins were first discovered as transforming products, so that's why they're called proto-oncogene products. They can cause the cells to become abnormal or transform once they're infected, and that results in cancer or tumor formation. So they were discovered as these transforming products. and when transfected into cells, cause them to become abnormal or transformed. And then this results in cancer or tumor formation. Okay, so we know these are monomeric, so they're single chain polypeptides, about 189 amino acids in length, so not very big. They're bound to, just like we talked about with the lipid modification of the trimeric G proteins, these are also bound to the plasma membranes by lipid modification of the C terminus. And they bind GDP and GTP, and they act as GTPases, just like the alpha subunit did for the trimeric G proteins, right? So they are, like we said, they're involved in tumor formation. And what happens in these cases is that there's some kind of somatic mutation that leads to their permanent activation. So there's two ways this can happen. Two ways these mutations can lead to permanent activation. What do you think can happen to lead to permanent what two mechanisms would lead to permanent activation? Chetan, if you have any idea, you can just raise your hand or speak up. So what I'm asking is, what two ways do you think that we could lead to permanent activation of these RAS proteins? When you think about the way G proteins are activated.
Maybe the GTP gets harder to be eliminated, so that it's like stopped.
Right, so what do we call that when we eliminate the GTP? What's it?
Dissociation.
Or
Hydrolysis.
hydrolysis, right? Yeah, so one could be that the mutation inhibits the rate of GTP hydrolysis. I need a new tongue today. So remember when we talked about those constants, the catalytic constants and the dissociation constant? So do you think this would decrease or increase one of those constants, and which one do you think would be affected? The catalytic or the dissociation constant? What?
Catalytic.
Catalytic, yeah. And would it be increased or decreased? Decreased, right? So this would decrease the K catalytic, right? Okay, so that's one way. What's the other way? So you decrease the rate of GTP hydrolysis. What else could you do?
Continuous binding of the ligand to the receptor.
You can what? I'm sorry?
Continuous binding of the ligand to the receptor.
Continuous binding of the ligand to the receptor, sure, but that is not a mutation in RAS, right?
Yeah.
So we're talking about mutations in RAS. Yeah, if you had a mutation in the receptor that caused the ligand to stay associated with the receptor, that would be true, yes. But we're talking about RAS now. What else could we do? So we mutated the rate of GTP hydrolysis so it stays on longer. It doesn't dissociate from RAS. What else could happen? What's the other side of that? What else has to happen for GTP to bind?
GDP should dissociate.
GDP needs to dissociate, right? So what would enhance the activation of RAS if we're talking about a mutation that causes constant activation?
Higher dissociation of GDP, so
Right.
KD will increase.
Yes, exactly. So mutation enhances oh. Yee. Rate of GDP dissociation. So we would increase the dissociation constant. Right. Okay, yeah, so these are the mutations that are present in tumors that lead to permanent activation. Okay, but not all of them, all of the forms of RAS are mutated. Actually, the normal protein is known as CRAS. Right? And this is normally, it's part of signaling pathways that are involved in cell differentiation, cell proliferation, and normal cells need that as well, not just cancer cells, right? Hold on, no space. Differentiation, there you go. Okay. The other three here that are shown, HRAS, KRAS, NRAS, these are all mutated forms. And all of these inhibit the rate of GTP hydrolysis. So they allow GTP to stay bound longer. So that the RAS remains active longer. Okay, so then just looking at these abnormal proteins, if we go to figure 411, which is on page, what page? I'll find it. 411. Is this the right one? This isn't the right. Wait, hold on. Sorry, I don't think that's the right one I was looking for. Give me a second.
Page number 95. It is
Excuse me?
It's 411, right? So page number 94. It is part 1.
Separate
Chapter
binding
4,
sites
part-
for. Yeah, but this is the alpha summary. I was looking for the similarities. Why do I have that one? HRAS, KRAS. I think that's correct. I think I have the wrong figure there. Hold on a second. Yeah, it's actually should be first 415. Sorry about that. On page 102. So it just shows you the primary sequence of RAS and the mammalian RAS proteins. These are all mammalian, these H, K, and N. And they're very close. The first 164 amino acids are almost identical, right? And the C-terminal segment is very divergent except for one amino acid. And so it just tells you that the different sequence of the motifs and what they're important for. So, for example, the amino acids, and this is not to memorize, it's just for interest, but the amino acids, I don't know what numbers those are. It looks like, I don't know, like 11 or something, till whatever. It just shows you the amino acids in the first row that bind the guanine, not nucleotide bindings that are important for that. And then they have the effector domain, and then interaction with GEFs, and then where it's post-translationally modified, where we talked about those those molecules, those that they're modified so that they can bind to the membrane, right? So it just shows you that they're very similar. 85% similarity in the first 165 amino acids, and then the C-terminal 20 amino acids are heterogeneous except for one section. Okay, so just showing you that they're very similar. So similar.
Then so the mutation occurs in the last part only.
The what?
The mutation
Yes.
occurs in the last part only.
During post translation.
Like in the last the mutation, which means like the H, K, and N, RAS.
Yeah.
Like they are basically different in the last part only, other than they are very similar to each other.
That's okay. Well, they inhibit the GTP ACE activity, so they're going to have mutations. The red are the oncogenic mutations, so only in those the guanine nucleotide binding, which makes sense, right? Because it's causing the GTP to bind tighter and not being hydrolyzed. So it's going to be it will be in the first 65 amino acids. Those are the mutations. Similar in first, except for the mutations. Are you finding something that's says different?
No. I mean, we're just talking that like maybe the H, K, and N, it's written there that they are all similar till 161
Yes.
or 164. Just the difference is in the heterogeneous part.
Yes,
Yeah, they,
but
like,
that's
the
not where the
16
mutations are.
The mutations are early, but they in their like with respect to each other,
Yes.
they are only different in the heterogeneous part, like not the starting part. The mutations in the earlier part, that's right, but we are talking in comparison to the H, K, and N.
Oh, right. Yes. Right. Yes.
There's the second line writes that at 186 cysteine residue, the C-terminal segment is highly divergent.
Right. Yes.
So they are different in the last area.
Yes.
With respect to each other,
Yes.
not with the mutation. The mutation is the early part.
Yes. That's right. That's right. Yes. Yep. Good. And then so there's also other families that are related to RAS. Just to be aware that there's this Rho family that constitutes RAC and Rho. They also act as GTPases, RAB and ARF. There's a lot less known about these, but there's a lot more known about the RAS protein. Okay. So then on table 4.5 and 4.16 talks about the 5 domains of the monomeric. So we're talking about the different domains of these monomeric G proteins, GTP binding proteins, right? So basically there's 5 regions that they call G1 through G5. And it's really interesting, actually, if you look on figure 4.16, it shows you that I don't know what the EF2 is, we're just going to ignore that for now. But if you compare between RAS and G alpha subunit, you can see that the G alpha subunits have these same 5 domains as the RAS. So there is homology between alpha subunits of the trimeric G proteins and the monomeric RASes. And then it tells you in table 4 or 5 what these different sections do. So the G1 residues, it's just like 7 residues. They bind to the alpha and beta phosphates. In G2, they bind to magnesium and to the effector. G3 also binds to magnesium, and I don't know what glycine does here, but glycine binds to the gamma phosphate of the GTP. G4, it confers specificity for guanine over other nucleotides, so that's probably one of the more important domains, so that GTP will bind rather than other like ATP or CTP. And then G5 is the guanine base recognition site. So just the different domains of the proteins, again, just to be aware of them. And then finally, just like we saw with the alpha or the trimeric G proteins, they need to be post-translationally modified in order to bind to the membrane. And without these lipid modifications on these post-translational lipid modifications on RAS, they cannot be activated. And they show you in table 4.4. Again, this is not important what kind of modification occurs, what kind of if it's farnesyl or palmitoyl or whatever, it doesn't matter. They have to have this post-translational lipid attachment to help them attach to the membrane, or else they will not be active. Okay, so at this point I want to take a break, and I want you all to look up simvastatin and its interaction with RAS, and just take like 10 minutes to look this up. Simvastatin, it's all right here. So just do a search and see what you can find in the literature for like the next 10 minutes, and then we'll come back and talk about what you found, see how simvastatin interacts with RAS. And you can talk, you can work together. Sorry, Chetan, I mean, Okay, so let's come back together. Talk about what you found. Okay. So how does first of all, did you find anything where simvastatin can regulate RAS? Yes? Yes.
Yes.
Okay, so we know it can regulate RAS. I guess we should back up and ask what's the mechanism of action of simvastatin and what is it used for?
It's used for decreasing lipids,
Mm-hmm.
for treating hyperlipidemia.
Anything else that you prescribe it for, Matt?
That's the only thing I've seen it prescribed for.
Okay. All right.
Off the top of my head.
Right. So high cholesterol, right?
Yeah.
Give them some statins.
Yeah.
All right. Okay. So, and then we said statins can regulate RAS. So what can simvastatin do to RAS?
Mostly when it's decreasing, reducing the expression of rice.
Decreasing the expression.
Decreasing. What I have seen is like once it increases, it causes like it's important in cancer progression and metastasis.
Mm-hmm.
So there were studies which showed that simvastatin is reducing RS protein,
Mm-hmm.
leading to improvement in lung cancer, endometrial cancer, even in the breast cell invasions. And it has also seen that simvastatin can decrease mutated forms of RAS as well,
Mm-hmm.
like HRAS, which we discussed
Mhm.
earlier. So, what I have seen is that it has decreased RAS all over. Like,
Okay.
I have not seen anywhere that it's increasing the RAS.
So simvastatin has this. We just saw that KRAS requires that post-translational isoprenylation,
Right.
and only then it can function. So simvastatin arrests that by depriving KRAS of the isoprene units that it
Right.
requires.
Right.
Basically, the farnesylation and geranylation,
Mhm.
it stops that.
Right.
And another thing that I saw was because this happens, it also I don't know what was that, but it kind of stops the cell cycle at G1
Mm-hmm.
by, I don't remember like how, but then it stops the cell cycle, like it arrests the cell cycle phase at G1 in cancers, mostly because of the it scavenges the isoprene units.
Right.
Which is
So,
fine.
and RAS is a driver of cell proliferation, cell
Yeah.
division. So, by inhibiting that, you can inhibit the cell cycle at G1. What did you find out? Anyone find anything different or anything new to add to that?
No, mostly what Muslim and Vivek says, like since all statin blocks the HMG CoA reductase activity, so the target was to prevent mevalonate production, which is a precursor of cholesterol. But from mevalonate to cholesterol, there are other stuff that mevalonate converts into, which is you saw like geranyl, farnesyl, pyrophosphate. So what farnesyl pyrophosphate is required for is the activation of RAS. So when statin blocks the HMG CoA reductase, mevalonate production stops, and so is farnesyl pyrophosphate. Thus, RAS is not activated. And then I found a paper where it says that if RAS is inactive, the signaling downstream effector is also like blocked, which is ARC1 to AKT mTOR, and thereby it induces apoptosis through suppression of GGPP biosynthesis.
Okay.
That's the
Okay.
Green
Yeah.
and green and
Yeah. So yeah,
So, yep.
same.
Same.
And what I found was not only just inhibiting the cancer, but also inhibiting the EMT by
Oh.
uh,
Yeah.
I don't know, the blog is directly saying as well. So while they're describing was when the EMT is happening is through the RAF, MAC, and ERC the signaling, then the downstream was EMT outgrowth. So by inhibiting the HMG the HMG,
CoA
yeah,
reductase, yeah.
the reductase. Also, another the inhibition was they loaded like directly to probably through this last lab all the way to the arc probably that and downstream EMT as well.
Yeah. So EMT is what?
Um,
Do you know what that stands for?
transition.
Epithelial to
Yeah.
mesenchymal
Yeah.
transition. So when some cancer cells start out as epithelial, that's their phenotype. So they're rounded and not very spindle-like. So they stay in situ. They stay in place that when they're epithelial in nature.
Right.
So if you can keep them in that form rather than transitioning to mesenchymal, which are very spindly, and they migrate, they move. The mesenchymal cells are the ones that cause metastases, right? They leave the initial spot of cancer and move to other spots. So if you can inhibit that EMT of cells that start, cancer cells that start out epithelial nature, you can keep them in place, and then they're also easier to treat. They're not as resistant to treatment as the mesenchymal cells. Actually, one thing that our lab is interested in, I don't know if you caught it in Asif's seminar, is the reversal of the EMT. So MET, taking the mesenchymal cells, because there are cancers like triple negative breast cancer.
Yes.
That's what we focus on. They already start out very aggressive and mesenchymal in nature. They're already like as soon as they're found, we know they have a high propensity to metastasize. So we're trying to take those cells that are already mesenchymal and reverse them back to epithelial so that they'd be more treatable.
Ma'am, I have a question.
Yeah.
Like, it might be not in the domain of the
Okay,
class, but I would
alright.
like to ask it.
Sure.
Is it fine, ma'am?
Yes,
Yeah.
that's fine.
Like, ma'am, when I saw that presentation and there was a class that the heading was is the reversal of metastasis.
The what?
The reversal of metastasis.
Yes, right, okay.
Ma'am, is it like quite right to say that the reversal of metastasis, or if we are saying that it's from EMT to MET rather than saying then reversal of metastasis? Because metastasis is something migration.
Yeah.
Once it's done, like what benefit you will take when you, like, is it going to the back to the primary site, or it is just staying there and converting to epithelial, and then we can treat them over there and they cannot migrate further.
Right. Yeah, so it's not 100. So the question is Chaitan in that class?
Yeah.
Yes.
So when the student in my lab, Asif, gave his presentation, he said, I think the title was related to inhibiting metastases, right?
Reversal of metastasis.
Reversal of metastases. Wow, that's a lofty goal. So
That's my
so
question
yeah,
as
so
well.
I think so. When he came up with that title, was to grab people's attentions, and I'd say it's 50% correct, right? Because if we get the cells, we're hoping to transform both the cells from mesenchymal to epithelial, whether they've metastasized or not. So if we catch a patient early enough and we can keep the cells in situ before they've started to migrate, then yes, that could inhibit
Inhibition of the metastasis.
Inhibit
That's
metastases,
more-
not
reversal
really
of the metastasis.
not reverse it. Like, yeah, so we're not, once the metastasis has occurred, the cells that are there, if we can reverse them to epithelial, so one of the common sites from breast is to the lungs, right? So if we can reverse those cells in the lungs from mesenchymal to epithelial, they'll be easier to treat in the lungs, but they're not going to migrate back to the breast. Yeah, and that tumor's going to go away. No. yeah. So it's a little bit of a misnomer, but yeah.
I think he meant reversal of EMT.
Yes. Yeah, pretty
What?
much.
That would be
That would be a
more
more
accurate.
accurate
Yeah.
title. Yeah, I agree.
The reversal of metastasis catches a lot of.
Oh yeah, like what?
Yeah.
Like, yeah, but even the reversal of MET is pretty amazing because there's not many
Yeah.
things we know that can do that actually.
It's still very like difficult thing to achieve, right?
Yes, yes.
Inhibiting the EMT to well, either inhibiting the EMT or converting it to MET. Like,
Right. So there's lots of so what this is totally a tangent, but might as well like promote my research. So there's a lot of things we know that will block the EMT, so the epithelial to mesenchymal transition. They're like ERKs and PI3 kinase and all that stuff. But there's, as far as we know, there's very few small molecules that have shown that they can reverse the mesenchymal back to epithelial once it's already occurred. So that's why he didn't show this unfortunately, but what happened was we had a lead compound before Asif joined the lab. We had a couple lead compounds. We were focusing on ERK5, our favorite kinase, and we had a couple lead compounds that showed selectivity for ERK5 over other signaling pathways, right? So we sent them to our collaborators in New Orleans who have do our animal research for us, and because we don't have the animals housed here, and they were just going to first verify what we saw in cells, just in triple negative breast cancer cells. So they just wanted to, verify what we saw. So they plated their cells, treated with our inhibitor, and then they were going to look for ERK5 inhibition within 24 hours. Well, hurricane hit New Orleans. Yeah, hurricane hit, and so they weren't allowed in the lab for a week, just because it was so bad. And Tulane's right there. It's right in like on the coast, so they couldn't get into their lab. So they got in a week later, and most of us. This is actually a really good story for students because most of us would probably throw that experiment away, right? You'd be like, oh, okay. We were supposed to look, harvest these within 24 hours. Well, they decided to let's just see what happened. Let's look under the scope, see if the cells are even still alive. And what they noticed is that these very spindly, aggressive cells, not all, most of them now looked very rounded and not like the normal phenotype. So they did some experiments looking for epithelial markers versus mesenchymal markers, and it indeed did reverse those cells from mesenchymal to epithelial in nature. So that was very cool. Yeah, it was very serendipitous, right? Because if that hurricane, not that I want the hurricane to strike anytime soon, but not if that hurricane didn't strike and if they would have just thrown them away. So don't throw away your experiments. Always take a look and see what's going on, even if you think it's a, quote unquote failed. Something interesting might pop out of it. So we were lucky they looked at those cells just to see, because that's very cool. So that's what led us then down. Instead, we were first focused on proliferation, migration, tumor size, tumor genesis, tumor growth, all that stuff. But then when we saw that, we're like, oh, we should really focus on this EMT, MET question. So that's kind of how we shifted gears towards that. And so that's where we are. One of the places we are. So cool. So all right. So all that to say that simvastatin can regulate RAS. It can decrease the expression. So does it decrease the expression of RAS, or does it decrease its ability to bind to the membranes?
I think bind to the membrane, like activation.
Okay, so not expression. So this should be activation. That's what I thought.
Post-translational activation of the protein.
Post-translational. That's even better. Post-translational decrease or inhibit. Post-translational modification. Right? Okay, good. So did you find anything that relates this to its main mechanism of action for hyperlipidemia or anything else that can help these patients? Yes?
So in hyperlipidemia, you have high blood cholesterol, and cholesterol. If you see biosynthesis of cholesterol, it has those single isoprene units, unsaturated isoprene units. They come together and form ultimately give a cholesterol structure.
Yes. Right.
So HMG-CoA reductase is inhibited, and you do not have you basically you cannot assemble these isoprene units to form mevalonic acid,
Right.
and hence like the further biosynthesis of cholesterol is inhibited.
Mm-hmm.
Cholesterol and other forms of lipids as well, they're inhibited, like
Right.
sphingosines as well.
Right.
So I believe that is one reason that it is used in hyperlipidemic patients.
Yes, yes. But is there any other benefit for these patients that RAS, I don't know, I'm just asking if you found anything, that RAS activation is also decreased? What do you think? Did you see anything when you were... I just wondered if you saw anything. Anyone? No?
I have a
Yes.
question.
Yes.
Yeah, I was thinking about I have this question in mind because I saw one paper that showed the expression of RAS was dose dependently decreased with rosuvastatin and simvastatin. So I was trying to connect whether because we've been talking about RAS activation, not expression. If there could be some link, because they showed Western blots and not the activation per se. So
Just what Western blots? I'm sorry.
RAS protein Western.
Oh,
blot.
expression to see if expression is
Yeah, so
decreased.
expression was decreased.
Expression was also decreased.
Yeah.
Oh, so it's expression and activation. Okay, we'll put that back in then. There we go. Expression and activation is decreased. Okay. I don't know. I mean, I don't know how it would help people that RAS was activation was decreased. Maybe something. I don't know what. Maybe some gene that is activated by RAS downstream that is involved in lipid production. I don't know. Maybe. I have no idea. But just, I mean, obviously the main thing is that they block the cholesterol synthesis, right? Formation. So that's the main thing. But I just wondered if, inhibiting RAS would also have some beneficial effect for these patients, possibly. There's something else I wanted to think of. Also, so you some of you have seen this in when you just did this brief search. You saw that it was used to treat some cancers, right?
Yes.
Yeah. So it kind of makes sense, but it also, I have a question, and I don't know if it came up in the papers, but okay, it makes sense because you inhibit RAS, so you're going to inhibit proliferation and differentiation, but proliferation for cancers, right? So, but what the C-terminal region we looked at is where this farnesylation occurs, or whatever, where the lipid modification occurs, right? But the mutations occur in that first 165 amino acids that cause RAS to be constitutively active. So I'm wondering, how is the statin working in that case, right? I mean, yeah, we need RAS to be tethered to the membrane in the normal signal transduction pathway, right? But if you have GTP permanently, or like more permanently bound, it's not hydrolyzed as quickly, right, on these constitutively active RASes. How would the statin affect that? I guess it would have to be in cancers where RAS is not constitutively activated, because there are some cancers where RAS is not the main player. I think it would have to be in those cancers, right? I don't know. Was it was this in a cell line, or was this in patients when you were talking about lung cancer?
It was cell line. The lung cancer was cell line.
It was a cell line.
Yeah.
So I wonder if that cell line has constitutively active RAS or not. I mean, I would imagine it couldn't, because if it was constitutively active, it wouldn't matter. Would it necessarily matter if it was tethered to the membrane? I mean, because it would have the GTP bound. I don't know. Just a thought exercise, just trying to think about it. Does it say anything about those cell lines? Quick, what are they? What's the cell line?
Lung cancer.
Yeah, what's the name of them? Is it like HT something, are they? Or is that breast cancer? I don't know.
The paper I found, they took it from tissues from patients
Ah,
after
okay.
surgery.
They actually took it from
Yeah,
tissues.
lung cancer
Yeah,
patients.
and is that type of cancer, does it say that RAS is mutated in that cancer? Because it's not mutated in all cancers, it's mutated in more highly aggressive cancers. So just wondered. It's okay if we can't find it. I would imagine it couldn't be constitutively active, right? Does that make sense to you? Because if it was constitutively active, the simvastatin wouldn't be able to inhibit it. Unless, if it's not tethered to the membrane, whether it has GTP bound or not, if it's not tethered to the membrane, if it gets degraded quickly or, I don't know, something.
So Dr. Kavanaugh, what
Yeah.
do you mean like when you say constitutively active?
So constitutively active means that it's active all the time. It doesn't require a ligand binding to a receptor. It doesn't require, the exchange of GTP for GDP, because GTP is just bound all the time. Because these mutations, like we said above here, right, they inhibit the GTP hydrolysis, right? We said that, right? Oops. Right here.
Right.
So if GTP can't be hydrolyzed, then it just stays there, and those are constitutively activated.
Yeah, this article is, I think, the same one. There's one. Normal and lung tumor tissues obtained from each sample were exposed to simvastatin, 2.5 to 30 microgram, or rosuvastatin, and Western blot analysis was then performed. And they
Okay.
document the increased expression of proteins MMP2, MMP9, NF, K-beta, and P65 in lung cancer tissues with respect to normal tissues. In the malignant tissues, simvastatin and rosuvastatin significantly and dose-dependently reduce RS proteins.
Well, okay, so if it could then just reduce the amount of protein so that even if it's constitutively active, there's not a lot of protein there to be constitutively active, right? I guess that could happen.
Yeah.
Okay. All right. Anyway, that was just a small exercise. Okay, so these GTPases, we have a lot of them. There's hundreds of monomeric GTPases, and they were identified by screening cDNA libraries for RAS-like proteins. So on table 4.6, which is on page 106, it shows you these different family members. We talked a little bit. There's RAS, but there's also the Rho, RAB, and there's these ARF and RAN proteins. And it shows you that the functions of these different proteins. So for example, on page 106, if you can get there, it says RAS. We already know this, important for cell growth, proliferation, and differentiation. The Rho family is important for the cytoskeleton and cell proliferation. RAB is important for vesicular trafficking intracellularly inside the cell. RAN, so some of these I've never really heard of that much, but this RAN regulates transport between the nucleus and cytoplasm.
Yes.
ARF is vesicular formation and activation of phospholipase. I think that's phospholipase D, PLD. And then these other ones that I've never even heard of, the CUR, REM, RAD, they're for cytoskeleton, cell shape, and voltage-dependent channel regulation, right? But so just important to know that there's RAS that we know a lot about, and then there's Rho and Rab that we know a little bit more about, and then some that we're still learning a lot about. And then you can see that some, obviously, of these have more than one function, and their function is going to be determined by what cell they're expressed in or where they're localized. And then I had figure 419 to look at. Yeah, so this just shows some of the influences that RAS can have. It can act, there's some of the effectors are PI3 kinase, RAF, which is upstream of the ERKs, some of the activators of the RAS, GTP, and some of the inhibitors. So then we talk about these RAS gaps, and trying to remember what they're negative regulators of RAS, and they accelerate the GTPase activity up to five orders of magnitude. So these accelerate. So that means it's going to increase K catalytic, right? Right? The catalytic GTPase activity, yep. Okay, good. All right, and so that makes sense that it inhibits the myogenic activation of growth factors. It also makes sense that they're negative regulators of RAS. So on in page 421, it shows how this kind of happens. So it's just showing some of the amino acids in the active site of RAS and its interaction with the RAS gap. So they did some experiments. So when these were first discovered, they did experiments to see whether they actually bound to the RAS or they did not, and they found that there is some stoichiometric reaction between the gaps and the RAS here, right? And it shows you from the the RAS gap, there's an arginine figure. So this R1 is an arginine residue, and that's what causes the interaction between the gap and the RAS. And the gap basically neutralizes two negative charges on the GTP that's bound to the RAS, right? And it also needs this secondary arginine residue, this R2, to stabilize this interaction of R1 with the RAS. And then what it does is by interacting with the GTP here is it opens up the RAS protein to expose this glutamine residue, this Q61, and in doing so that allows the water to come in and allows the hydrolysis of GTP. Okay, so it's basically a few amino acid residues that allow this GTPase activity to occur. Look at that Q61, that glutamine 61, and go back to the figure that we looked at for the structure of the RAS on page 102 and that 182 amino acids, and look at Q61, that glutamine. That's a red. That's one of those residues that are mutated, right? Yes. So if it's mutated, right, it wouldn't allow these this gap to bind, or it would allow the gap to bind, but since there's no glutamine there that's mutated, it wouldn't allow that hydrolysis to occur, right? That's pretty cool. All right. And then we also, so we have RAS gaps that can accelerate the GTPase activity, and then we have these guanine nucleotide exchange factors, or GEFs, that can catalyze the exchange of GTP for GDP. And so the way they figured out this should be RAS here. The way they figured out the function of these GEFs is that they would express these G protein coupled receptors in insect cells that do not have beta adrenergic. They use beta adrenergic receptors, for example, and this is figure 424. Figure 424 on page 118. And it shows you that they express these beta adrenergic receptors in insect cells, and then they saw that, they got some, without the receptor expression, they got some basal cyclic AMP. And then they expressed the receptor and they got a little bit more cyclic AMP. But then when they put in the agonist, they got some enhanced generation of cyclic AMP. And then what they did is they just kept adding components to the cell. So in D, they added the alpha subunit of the G protein to show that they could increase the affinity of the receptor for isoprenaline, and then also increase the generation of cyclic AMP. And then finally, they got the highest affinity and highest generation of cyclic AMP when they put all the components, the alpha and beta gamma subunits, along with the receptor into these cells. So this just gave them an idea of how the components work together. Okay. So, what was that? I'm sorry, there was one thing. Oh, one other thing I wanted to talk about is that on page 119, figure 425, 4.25. This is a nice summary, kind of, we generally think, and how we teach it, and this is still how I teach it to the PharmD students, so we generally think there's a receptor with a ligand, so in that number one, ligand with a receptor to a G protein to an enzyme, right, to a effector. But really, all these other situations can occur too, making these G proteins even more diverse, right? So you can have two different receptors activating the same G protein, leading to the same effector, or you can have one G protein that activates different effectors, or you can have a receptor. This is the most interesting to me because we don't often think about this. You can have a receptor that switches between subtypes of G proteins. For example, they say in the figure legend there, the adrenergic receptors can switch between GS and GI, and then you'd have different effectors downstream. And then the final example 5 is two different receptors, two different G proteins, but one effector enzyme for them. So I think that's a really neat figure that pretty much summarizes, other than the other regulators like the GAPs and the GEFs and stuff like that, but it summarizes how diverse these can be, right? How you can have different G proteins activating different receptor effectors, or you can have the same G protein activating the same effectors. You can have two totally different receptors coming together to activate the same effector from different G proteins. And I think, to me, the most interesting is some receptors actually switch which G protein. And I guess that mainly depends. I haven't, did a deep dive into that subject, but I would imagine it depends on the cells that are expressing the receptor, the concentration of these G proteins in those cells, right? Things such as that. Okay, great. So the next thing that we'll start on Thursday actually is we're going to talk about the enzymes and the different subtypes of adenylate cyclase and phospholipase C, and look at their structure a little bit on Thursday. Before we end, though, what I wanted to say is I have not yet talked to Dr. Johnson yet about an exam versus not an exam, but I plan to talk to him before Thursday, and I think we'll do some kind of take-home, like summarize an article or something, just to get you writing too. You did a few presentations here, which was good, but we also want to enhance your writing and your scientific writing through these courses. So I'll talk to him and I'll come up with some kind of take-home exam for Thursday that we could have, I don't know, however much time you need, a week or 2 weeks to get it done. Okay?
Yes.
And I think Dr. Johnson comes back next week, I believe.
Tuesday.
Tuesday. Yes, not Thursday, Tuesday next week.
Because written 3 stars on it, and it was written downside that it's not confirmed.
Oh yeah, I have to talk to him because he had. I think that maybe Tuesdays didn't work for him. One of the days, Tuesdays or Thursdays, did not work for him, so he was gonna arrange with you when would be a good time to do the second class because he couldn't make it work on or double up on a Thursday or something. I know, sorry, I know it's a lot of time, but we'll see how that goes. Okay, I'll talk to him about that as well. But